Tantor Audio, a division of Recorded Books, presents The End of Genetics, Designing Humanity's DNA by David B. Goldstein, narrated by Christopher Douyard. Publisher's Note: in this audio edition, you will hear references to visual elements present in the print and ebook formats of the book. Introduction. My career has been focused on identifying the genetic changes that cause disease in individual patients, and on trying to use those genetic insights to develop effective treatments that target the precise causes of disease. Over the years, this kind of work has been called personalized medicine or genomic medicine, and now it is increasingly referred to as precision medicine. Whatever name we've given it, it has been both satisfying and frustrating. Genetics research has progressed more rapidly than many geneticists, myself included, had expected. As you will learn in this book, from devastating genetic diseases of childhood to later onset and presumed more genetically complex diseases, such as amyotrophic lateral sclerosis, ALS, kidney disease, and heart failure, the mutations responsible for disease are being identified in ever-increasing proportions of affected individuals. Treatments for these genetically resolved diseases, however, are lagging increasingly far behind the genetic discoveries. A recurrent theme has been waves of growing enthusiasm that new, precisely targeted treatments are just around the corner, then followed by crushing setbacks. We are currently in a period of renewed optimism about precision medicine, driven above all by stunning scientific advances in genomics, gene editing, stem cell biology, and pharmacology. But it is important to appreciate that we have been here before. In the 1990s, many people thought that gene therapy was poised to dramatically improve the lives of a large number of patients suffering from genetic diseases, a lot of which had been newly diagnosed through our growing understanding of the human genome. Numerous trials were initiated in which viruses were used to introduce functional versions of genes that patients lacked because of inherited mutations. One trial that was destined to change the field was initiated at the University of Pennsylvania and focused on a genetically transmitted disease caused by a deficiency of an enzyme called ornithine transcarbamylase, OTC. The disease is due to mutations in a gene on the X chromosome and affects mainly males, who have only one X chromosome. The mutations impair the function of the enzyme, which helps to break down nitrogen, and people with OTC deficiency can develop toxic levels of excess nitrogen in the body in the form of ammonia. In 1998, a young man with OTC deficiency, Jesse Gelsinger, enrolled in the trial and was treated with a modified cold virus. The modified virus was meant to introduce a functional version of the OTC gene to Jesse's liver, and therefore restore the normal processing of nitrogen, and thereby eliminate the toxic excess of ammonia. Instead, Jesse's body mounted an unexpected immune reaction to the virus used to introduce the functional OTC gene. Within a day of the infusion of the virus, Jesse showed signs of jaundice and progressed to multi-organ failure. Within 5 days, he was dead. Later analysis suggested that the virus ended up being distributed much more broadly throughout Jesse's body than expected, and contributed to a devastating and ultimately fatal immune reaction. The rapid growth in gene therapy trials came to an immediate end, and it would take more than 10 years for public trust and enthusiasm to be regained. This reminder of the challenges in developing genetically targeted therapies is not intended to imply that the current enthusiasm about precision medicine is unjustified. Indeed, scientific progress in this area has been truly remarkable, and this scientific progress led to increasingly important advances in treatments targeted to underlying causes of disease. Some of these you will learn about here, and some you will have read about in the news in recent years. But these advances are not coming anywhere close to keeping pace with our exploding knowledge of the underlying genetic causes of disease. I have been concerned about the implications of this sharp discrepancy for many years. A decade ago, I wrote in Nature that the explosive growth in the identification of genes responsible for disease, coupled with modest progress in developing treatments for those diseases, would inevitably lead to increasing interest in ensuring that the genomes of children do not carry such mutations. Despite my being an enthusiastic advocate of, and, wherever possible, contributor to precision medicine, the past decade has only added to my concerns about how limited our options remain to effectively treat genetic diseases. Not only do we lack effective treatments for most single-gene disorders, we now know that the challenge is even greater than finding a treatment that can undo the direct effects of the disease-causing mutations. The reason lies in the complexity of human biology. Let us imagine the discovery of an entirely safe gene therapy treatment for a genetic disease. That is, we will envision a therapy that is capable of introducing a replacement for the faulty gene into exactly the cell types that need it. Let us further suppose that the replacement gene is expressed at the right times and in the right amounts, and that no immune reaction is engendered. Unfortunately, even this achievement, which is still very far away from most diseases, would not be sufficient in many cases. We now know that many human diseases have important developmental components. This means that the presence of the faulty gene early in life alters the developmental trajectory of the child, and even that of the fetus. For such diseases, even a completely effective replacement of the faulty gene postnatally would not represent a complete cure. For these reasons, while remaining committed to the paradigm of precision medicine, I also remain convinced that many of the most devastating diseases will ultimately be treated, not by precision medicine, but by the careful determination of the genomes of our children. If I am correct in this conviction, in the future, parents will be faced with complex and consequential choices about how they wish to adjust the genomes of their children. And, to make those choices, parents will need to understand the underlying science so that their decisions can be as informed as possible. What they will need to consider is not only what we know about human disease genetics, which the media are often eager to convey, but also what we do not know. Parents will also need to understand what can practically be done to adjust the genomes of their children, and what the consequences of any changes are likely to be. And because it is not an overstatement to say that the decisions that parents ultimately make will influence the future of our species, the purposeful design of the genomes of children is something that should concern us all, whether your reproductive years are before you or behind you. One conclusion I have drawn from these considerations is that, although human genetics has already advanced far enough to provide parents with information on which they may choose to act, key gaps in our knowledge make it very difficult to anticipate the consequences of actions likely to become possible. These gaps mean that any reproductive engineering that is performed beyond the most straightforward elimination of strongly acting disease-causing mutations will be performed without a complete understanding of the likely consequences of those changes. This is a prospect that I find deeply troubling, and this book above all represents my best effort to empower non-scientists to develop their own opinions about this most central question for the future of humanity. In response to this deep uncertainty, I have developed a thought experiment in reproductive genomics to help illustrate the kind of genome engineering that could be entertained in the not-too-distant future. Throughout the book, I will refer back to this thought experiment to help make clear that we will have the technological ability of making some kinds of adjustments to the genomes of children, without having a matching ability to accurately predict the consequences of those adjustments. You will be in a position to understand this thought experiment more fully later in the book, but as a motivation in what follows, consider the following possibility. All humans carry genetic variants that are very rare in the general human population. Some of those are brand new in each person, and some are variants that appeared first in their parents or grandparents, or in some fairly recent ancestor. These variants, which natural selection has not yet had the opportunity to examine closely and pass judgment on, as long as they are compatible with life, are often damaging to the function of the protein encoded by the genes they affect. Our simple thought experiment, then, is this. What would happen if a couple were to decide not to transmit any such rare variants? I refer to such an experiment as leading to the common variant human, in which every place in the genome where a rare variant might occur is replaced with the variant that is common in the human population. That is, a variant that natural selection has reviewed and judged acceptable. In preparation for this book, I asked a number of prominent geneticists to essay a prediction about this thought experiment, and found widely varying responses. I do not claim to know the correct answer to the outcome of this thought experiment myself. The main point is to convince you that we will be able to actually perform this experiment in the future, and that we will be able to do so long before we know with any confidence what the outcome will be. The title of this book is of course modeled on the title of Francis Fukuyama's book The End of History and the Last Man, published in 1992, an elaboration of his essay The End of History from 1989. Fukuyama argued that liberal democracy was a socio-cultural end state, and, with its ascendancy, the evolution of human social organization would come to an end. The experiences of the present, with an apparent march toward authoritarianism in many parts of the world, would suggest that this thesis is demonstrably wrong, as most political scientists would today likely agree. Yet, there were compelling reasons that Fukuyama made his argument. We can perhaps hope he will, in some way, turn out to be right. My title is meant in the same way. I do not mean that human genetics will end, but there are reasons we can argue that the form human genetics has taken until now will shortly come to an end. Here are some clear examples. Human genetics is today, and always has been, a lottery. We all carry mutations that can cause recessive diseases. For each one, there is a 50% chance that it will be transmitted to a child. If it is combined with another such mutation, the child will have a recessive disease. Beyond these mutations that are found in a child's parents, each child born will have its own collection of brand new mutations, which will appear in the sperm or egg that led to the child. For example, if we consider only point mutations that change one particular site in the genome from one thing to another, these things changing will soon be fully explained. Each human born today has between 50 and a few hundred brand new mutations distributed throughout his or her genome, with the exact number depending very strongly on the age of the father. If the child is lucky, these new, or de novo mutations will fall into parts of the genome that have little or no function, and will do no serious damage to the child's development and functioning through life. Each embryo conceived, therefore, is playing an unknown and deadly serious game of genomic Russian roulette, with parents implicitly hoping that all these new mutations will fall somewhere harmless in the genomes of their children. It is a game of chance that all couples through all of time have been obligated to play in order to have children. This is how we have reproduced, and how we still reproduce today. It is not how we will reproduce in some tomorrow, and that tomorrow will come. I believe that it is now certain that parents will soon, and increasingly, decide on the genetic makeup of their children in ways both large and small. When this begins on a large and systematic scale, it is going to constitute a whole new kind of genetics. In consequence, human genetics as it has been since our beginnings will be over. Of course, a new kind of genetics will replace it. What exactly this new kind of genetics will be is for us all to decide. And I believe that everyone should be in a position to participate in those decisions. I hope that this book will help. Chapter 1: The Future of Reproduction. The first decade of the 21st century marked a turning point in the relationship between society and the human genome. For the first time, it became possible to determine the genetic makeup of any person in a matter of days, and at a cost already within range for many millions of people. Even before this genomic watershed was reached, a movement had emerged to provide genetic information directly to consumers. In some cases, the offerings to consider included help to make more perfect babies. The obvious question upon reading such a claim is this: what would qualify as a more perfect baby? Until recently, this question was of primarily academic interest. Ethicists could debate the benefits and the risks, but there was no realistic prospect of systematically prohibiting the appearance of unwanted mutations in our children. For better or worse, the genomic engineering of future generations has suddenly become a very real prospect, and is therefore something, I believe, that society must urgently consider. There are two key questions about such reproductive genomic engineering that need to be considered and understood. What do we want to change in the genomes of our children? And what are we likely to be able to change? In terms of what we would like to change, in my experience, most people are comfortable with the idea of ensuring that children do not carry mutations that cause devastating childhood diseases. Indeed, there are already ongoing efforts throughout the world to reduce the transmission of severe childhood genetic diseases, such as Tay-Sachs disease. Many couples already opt for a procedure called carrier screening, which seeks to identify genes for which both mom and dad carry mutations that, when combined, would result in a severe childhood genetic disease. And many fertility centers offer the option of testing embryos for mutations that would cause such diseases, in order to select for transfer those that do not carry two such mutations, among other genetic testing options. But how far should these efforts go? Is it also appropriate to test for mutations that predispose one to, but do not deterministically cause, later onset conditions, such as Alzheimer's disease? And what about non-disease traits, such as height or eye color, or, indeed, traits such as sexual orientation, where the genetics is often only probabilistic, but where those probabilities can in principle be tested for, and parental preferences acted upon? Even if we restrict attention to disease, we face the question, what is or should be classified as a disease? These questions become particularly acute when we recognize that different people will have very different ideas about what they would like to see in the genomes of their children. And views about what is and is not a disease have changed in important ways over time. As one stark illustration, a certain minority of the deaf community has a preference for deaf children. What if a couple wants to use genomic technologies to ensure that their children would also carry the same deafness mutations that they themselves carry? In the past, professional geneticists could draw a certain comfort from technological constraints. As our ability to identify the mutations that make us different from one another grows, along with our ability to not only select, but, as we shall see, edit what is present in the genomes of our children, what we should and should not do will emerge as one of the defining questions for societies and individuals. The second part of the question about what we might change in the genomes of our children relates to what kinds of traits we are able to influence, and how much we can influence them. When the personal genomics movement was first gaining traction, it was heavily focused on common genetic variants, mainly for technical reasons related to how genetic studies at the time were performed. These common variants are conventionally considered sites in the genome where there is a common form and a minor form, and the minor form is observed in about 5% or more of the time. This represents a tiny fraction of the variable sites in the human genome. As you will learn in some detail through the course of this book, there are around 3 billion different positions in the human genome, and we know that most of these 3 billion sites vary in one or more humans alive today. As you will also learn in the chapters that follow, while we know that most of these sites will vary somewhere in the human population, we still do not know the consequences of the vast majority of that genetic variation. In the early days of personal genomics, there were two clear drivers of consumer interest: genetic ancestry and the results of genome-wide association studies, or GWAS. Genetic ancestry is just what it sounds like. Your genetics telling you something about where your ancestors are from in a geographic sense, and, sometimes, something about your relatives you may not have known you have. And a GWAS is a fancy and somewhat overstated way of describing a type of genetic experiment that allows the assessment of whether any of the common variants in the human genome influence a particular disease or trait. A typical GWAS might, for example, compare 1 million common variants between patients with schizophrenia and those without schizophrenia to see if any of them are associated with an increased risk of disease. As of this writing, many thousands of different common variants have been associated with hundreds of different diseases and traits, including, by now, almost all the common diseases and many other traits such as height and weight, skin and hair color, and even complex behavioral traits such as educational achievement, IQ, and sexual orientation. All of this can be reviewed using a catalog of GWAS findings. In the case of ancestry testing, consumers could learn something, albeit rather coarse, about the geographic origins of their Y chromosomes or their mitochondrial genomes, reflecting, respectively, their paternal or maternal ancestry. Or they might get a composite picture of a kind of average of the various geographic ancestries represented in their entire genomes, learning, for example, that their ancestry overall appears to be from some part of Europe, Asia, Africa, or the New World. Or they could learn that they are related, to some degree, to someone else who has also been tested. In the case of comparing their genomes against the results of GWAS studies of diseases and other rare traits, consumers might learn that they have a marginally greater or lesser risk of type 2 diabetes than the population average, or that they carry stronger risk factors for certain autoimmune or neurodegenerative diseases. In most cases, however, there has been no reputable advice that could be offered as a function of the genetic information gleaned from such genomic profiling. For example, one of the very strongest effects for any common variant in the human genome is due to the variation of the ApoE gene. Those who carry the risk forms of this gene have a greatly increased likelihood of developing late-onset Alzheimer's disease, but there is currently nothing that carriers can do to meaningfully alter their risks of developing the disease. This lack of clinical impact led me, in a New York Times interview in 2008, to sum up the entire enterprise as recreational genomics. Referring to the field as recreational was not only intended to convey that what was being discussed in the guise of personal genomics was of little to no clinical value, but also reflected a degree of exasperation that I, and many other geneticists, feel about how many personal genomics companies implicitly or explicitly oversell what they offer, and, in consequence, badly mislead the public about the nature of human genetics. Evidence of how personal genomics companies oversell and misinform is not hard to find. Sometimes it is subtle. Sometimes it is egregious. Alistair Moffat is a journalist and former rector at St. Andrew's College in Scotland. He is also the former chief executive officer of Britain's DNA, a now defunct for-profit personal genomics company that once offered consumers a variety of DNA tests. In the summer of 2012, Mr. Moffat described some of what Britain's DNA had discovered. He first claimed that a volcano 70,000 years ago blew itself to smithereens and destroyed all the human genetic lineages except for those of two individuals, dubbed Adam and Eve. He went on to claim that Britain's DNA has discovered a remarkable individual who has Eve's DNA, that they had found a genetic marker from Queen Sheba, that 33% of men in Britain carry the founding lineages of Britain, and that 97% of men with the surname Cohen share a genetic marker. The earlier description of what some personal genomics companies offered as recreational genomics can hardly cover excesses like this. An English geneticist, Mark Thomas, who is also involved with me in some of the work that provides the grain of science behind these absurdities, more accurately described them as genetic astrology. Astoundingly, Mr. Moffat's initial reaction to being challenged about the accuracy of these claims was to threaten legal action against Dr. Thomas and one of his colleagues, Dr. David Balding, a highly respected and talented mathematical geneticist. Mr. Moffat's science-free musings would almost be amusing if their potential consequences were not so serious. The egregious misrepresentation of genetics for what seems to be commercial gain reflects an increasingly complicated relationship between professional geneticists and a public eager to know what the vaunted genomics research might mean for them in terms of their personal histories and the diseases they might face. When the personal genomics craze got started, many professional geneticists felt there was little that was really useful for anyone to know from having their genomes profiled in the ways possible during the dawn of personal genomics. It was very awkward, however, for geneticists to insist that people have no reason to learn anything about their genetic makeups if they are interested in doing so. Insisting that people should not be interested when they clearly are reflects a degree of paternalism that many geneticists would be reluctant to embrace. The transition toward sequencing human genomes on a mass scale, however, moves us suddenly into an entirely new dynamic, much sooner than most geneticists were expecting, certainly sooner than I expected. We suddenly had the capacity to identify mutations of major effect wherever they are in the genome, and in whomever they occur. Already, this capacity is filtering through to the personal genomics companies, which are increasingly offering information about mutations that could really make a difference to people. For example, 23andMe, a generally much more sober personal genomics company than Britain's DNA, got itself into hot water several years ago by offering consumers information that has explicit medical relevance, in particular in testing for a panoply of disease-relevant mutations that might, for example, increase the risk of breast cancer, or that might cause an inherited genetic disease in children if coupled with another mutation in the same gene. The FDA ordered 23andMe to stop marketing their test because it offered medical advice without ever having shown that the test worked as advertised, leading to a lengthy negotiation between 23andMe and the FDA about the right way to convey such information. We are clearly now moving into an era in which getting things wrong really matters. Although I support the right of people to learn about their genomes if they wish to, I have little confidence that personal genomics companies, largely unregulated at present, can be generally relied on to provide accurate information. It should be clarified that the most significant concerns currently apply to mutations that have a very strong impact on disease, not ones with more subtle effects. These kinds of major effect mutations do, however, appear to be responsible for a surprisingly broad range of genetic diseases, though certainly not all diseases. Two examples make the new reality clear. A number of years ago, I began a research program at Duke University using a then brand new sequencing technology to try to find the causes of childhood genetic diseases that could not be resolved any other way. Bertrand Might was one of the first children we studied. Bertrand suffered from an unusual genetic condition that has many similarities to known disorders involving how sugars are added onto proteins, but with a striking distinctive feature. When Bertrand cried, he did not make any tears. Bertrand had what we call an undiagnosed genetic disease. My colleague, Dr. Vandana Shashi, an experienced and talented clinical geneticist, was sure Bertrand had a genetic disease, and equally sure that it was new. Nothing quite like it had been described before. The Might family had spent the first years of Bertrand's life shuttling from doctor to doctor so that Bertrand could undergo genetic and other tests to find the underlying cause of his condition, but with no success from this wrenching diagnostic odyssey. In 2010, Vandana invited the Mights to join a research study in which we planned to sequence the genomes of 12 patients like Bertrand and their parents, and to find all the mutations present in the portion of each patient's genome that encodes proteins, the most important part of the genome in terms of disease-causing mutations. 6 months after the Mights entered the study, we had the answer. Matthew, Bertrand's father, carries a very rare and destructive mutation in the N-glycanase gene, which encodes a protein that strips sugars off other proteins. But because Matt also has a normal copy of that gene, he is fine. Bertrand's mother, Christina, carries a different extremely rare destructive mutation in the same gene. But she also carries one normal copy of the gene and is similarly fine. When we sequenced this family, we found the mutations in Christina and Matthew and discovered that, tragically, Bertrand got both of those destructive and rare mutations. He does not make any functional version of this protein at all. When we performed this analysis, the responsible gene, N-gly1, was not yet known as a disease-causing gene. But we felt confident enough to diagnose Bertrand as the first patient with a previously unknown disease related to how defective proteins are removed from cells. We arrived at that conclusion, in part, because analysis of other human genomes suggested to us that people should not be found without any working copies of the protein this important gene encodes. This kind of approach, of learning what parts of the genome do and do not carry certain kinds of functional genetic variation, my colleague Andrew Allen of Duke and I eventually developed into a new formal tool for genome interpretation that we referred to as intolerance scoring, which I will describe in Chapter 4. The point for now is that we had looked through the entirety of Bertrand's protein coding genome and
Sample complete. Ready to continue?